Well, I think we can get started now. My name is Mark Musen. I'm from Stanford University in the United States, and it is my pleasure to welcome you to the next segment of what will be an all-day extravaganza regarding the NIH BD2K initiative. This morning you heard an introduction to the program, heard a little bit about work being done in machine learning in BD2K. In the next session, we'll be talking about issues related to metadata and the indexing of online data sets. And I will begin by presenting work from my own laboratory, which is, again, one of the centers funded by BD2K. So everything you'll hear for the rest of the day really will represent work being performed either directly by one of the BD2K centers or indirectly. My first trick is going to be to switch the projector. And that worked, so hold your applause until the end. What I'm going to be talking to you about is a project called CEDAR. CEDAR is the Center for Expanded Data Annotation and Retrieval, and it represents an initiative that we're taking on at Stanford University in coordination with partners at the University of Oxford, at Yale University, at Northrop Grumman Corporation, as well as a variety of other collaborators, all of whom are concerned with the idea of taking the data that we put online and making those data more accessible, more useful, more integratable, useful for all the good things that you heard about this morning. And in fact, this morning you heard the idea of fair data. You probably have heard the word fair data so much that you're probably almost sick of it by now. But I will not let this phrase go away, unfortunately, because one of our major goals in CEDAR is to make data more findable, accessible, interoperable, and reusable. And we believe that to do so really requires more than just being careful about the kinds of data that we put online, but it means we need to be very careful about the metadata that we use to annotate data. That means basically we need to be able to have experimental data available in open online access repositories. We require annotation of online data sets with adequate metadata that describe what the data are all about. For us, it means we need not just a description of the data, but we want to use ontologies to give us controlled terms so we actually know what we're talking about. And we want mechanisms to search for metadata and to find the relevant experimental results. BD2K, as you know, has put a lot of effort into search for online data sets. It's our belief that that's very important, but search is going to be fruitless unless the search engines can have metadata available to them that are reliable, that are standardized, and are accessible for all of the purposes that we've talked about today already. So in order to make data fair, first of all, we need to make sure that what exists in the world is described completely and consistently. And for us, that means being able to describe things clearly in terms of standard ontologies. Now, many of the online descriptions of the kinds of metadata that should be associated with data sets will make allusions to the idea that ontologies are important. We take it to heart that ontologies are absolutely essential for providing standardized terms that allow us to describe unambiguously what the data are that our data sets need to represent. In the case of CEDAR, we do this through BioPortal. BioPortal is a resource we've developed at Stanford, which is the largest compendium of publicly accessible biomedical ontologies. BioPortal now has more than 560 online ontologies. I don't say that having 560 online biomedical ontologies is necessarily a good thing, but it represents the state of our culture and the fact that there are so many proposed standards out there. BioPortal takes the position that we should have all of them included and have all of them mapped to one another, so if a user decides to use one, we can at least understand how that particular ontology might relate to other ontologies in the repository. But that's absolutely of first-rate importance to us. Secondly, we believe to make data fair, we need not only standardized terms to describe the elements of the metadata, but we need to describe the properties of the experiments completely and consistently. And for that, what do? Well, we know that metadata can be used for a variety of purposes, all of which are important to CEDAR. That means metadata that can tell us where the data come from, what kind of experiment generated the data, how the data were produced, how the data are currently formatted, where we can learn more about the data, what publications might be associated with the data. And eventually we'll want to include things in the metadata such as who has cited these data, who has re-explored the data set and created new discoveries. Those things are also going to be important to us. But it's absolutely critical that we have metadata that give us the ability to make sense of all of that. And we also need standards. We need standards for the kinds of things that we want to say about the data. We want standards for the language that we use to say those things. And we want standards for how we structure the metadata online. And we need a scientific culture that has religion, that actually believes that it makes sense to take the time and the energy to be able to create the kinds of metadata that will be most useful, not perhaps for the original investigator, but for the large community of investigators who ultimately will benefit because those data will become accessible and available online. And we know that we can do this. We know that in 2000, the microarray community was convinced that the only way that they could make sense of all their work would be to come up with a standard that would make it possible to talk about microarrays in a coherent manner. And what came out of this was a concern that all metadata for DNA microarrays had to explain what was the substrate of the experiment, what was the platform that was used, what were the experimental conditions. And from this we got Miami, which we now take for granted. So Miami gives us a list of terms by which we can think about what are the kinds of things that we want metadata about microarray experiments to say, that they can tell us what was the platform, how was it used, what do we know about the sample, what can we infer about the results. And we take it for granted that Miami is the way that we want to talk about these kinds of experiments. And what's really interesting, I think, in the biology community, as you all know, is that after Miami was proposed, there were a whole bunch of other successors. And Susanna Sansoni in her work has cataloged those successors, all of which deal with the minimal kinds of information that one wants to be able to express about particular kinds of biomedical experiments. What you see on the slide is an older version of Susanna's work showing a portal simply for the kinds of data or metadata that could be used to describe different experiments from different kinds of particular instruments. As you know, this has grown into something which is much more sophisticated that Susanna actually will be talking about later in the program. So having these kinds of standards is really important, so it gives us the ability to think about what we're going to say about data sets in a way so our search engines can find those data sets and actually interpret what data are there. But it's still not enough, because we seem, as investigators who have done everything possible to make our lives more complicated. For example, if one wants to submit data sets to GEO, doing so is really very, very painful. One has to use spreadsheets which are not well described, where the requirements are not well delineated, and try to fill in the spreadsheets with the relevant metadata, which kind of conform to the Miami standard, but are very, very difficult to fill out. And we know they're difficult to fill out because when you look at GEO, what you see is really a lot of junk. You see that when people are filling in these kinds of spreadsheets, the kinds of metadata that appear online, the kinds of metadata that we believe are going to tell us what our experiments are about, the kinds of metadata that search engines are supposed to rely on in order to find the appropriate data sets for retrieval, are obviously completely inconsistent, riddled with typos, and obviously don't give us the information that we need in order to be able to get access to information in a way which allows us to take advantage of those data in ways which allow us to combine data, to integrate data, to re-explore data, to do all the kinds of things that we expect to be able to do in the 21st century when data are made available online. And so to make data fair, not only do we need ontologies, not only do we need standards like Miami, but we need technology and we need a culture where it's actually a palatable experience to create metadata, and we can do so relatively easily. And that's what CEDAR is all about. So with that preamble, CEDAR tries to take all of those pieces, puts them together, and makes it possible for investigators to author metadata in a way which we think is convenient and which we hope will lead to an ecosystem where not only do we have more and more data online, but those data are fair because they are annotated with metadata where we can actually make some sense about what those metadata are about. So CEDAR can be viewed in this cartoon as a sequence of 3 different panels. In the leftmost panel, we're talking about actually authoring the templates that are going to be used to specify online metadata. In the middle panel, we take the templates that we've created in CEDAR and we fill them in with information about the particular details of the experimental data that are going to be put online. Ultimately, we can take those completed metadata specifications with the data sets and put them into some repository. But one of the things we also do in CEDAR, as you see in the rightmost panel, is we save the metadata that we author. That gives us the ability to search our local cache of metadata and identify how metadata may change over time and how experimental conditions may change, and things that we may learn about the nature of science from the metadata. But also what's important for CEDAR is that because we keep a local record of all of the metadata that people have authored with the system, we can use that local record to learn patterns in metadata and to use those patterns to inform, at the time that new metadata are authored, how we can take advantage of the patterns in order to ease the creation of new metadata. And I'll show you what that looks like in a bit. So CEDAR basically gives us technology where there are mechanisms to author templates that reflect community standards like Miami and all the others that are in the fair sharing resource that Susanna maintains, to fill out those templates, to encode experimental metadata, and to use that repository of metadata to learn metadata patterns and to facilitate the entry of new metadata. All of this links to the bioportal resource, which you've already heard about, which is this reference repository of online ontologies that allow us to use basically any ontology that has been designated as standard by anyone who uses it regularly in order to make sure that the metadata are filled in as completely and consistently as possible. So we look at CEDAR, we're basically looking at a web resource. CEDAR gives us the ability to have a group of users who have access to various aspects of CEDAR. It's templates, it's groups of templates that are shared by individuals, by laboratories, by larger groups. And if, for example, we want to be able to annotate data related to biosample, the NCBI repository that describes the samples that are used for a variety of different kinds of assays, and we're in particular interested in biosample human specimens, we can see we want to populate a template, and we get a template like this. So this is what CEDAR is going to lead us up to, taking us through the process of starting with a template and filling in that template with appropriate values. Now we do that in our middle panel, where we want to actually fill in the template, but we want to actually create that template. We're in the leftmost panel. What that means is we're going to be specifying for each field of the template what it's going to be composed of, whether it's going to be text, whether it's going to be numeric information, whether it's going to be qualitative information coming from some ontology. What we see here, for example, we're talking about tissues. And tissue is going to be a value, as you see at the bottom of the screen, so that little triangle icon, that is going to come from an ontology. So if we click on tissue, make it say it's coming from an ontology, CEDAR presents us with a variety of options that come from bioportal. We can see all of the ontologies in bioportal that may talk about tissues. And we can see Uberon, for example, at the top is an ontology that talks about tissues, but so does the mouse anatomy ontology, so does NIF standard, the Neuroinformatics Framework standard ontology, so does the Teleost anatomy ontology, which presumably you don't care about in biosample human, but Uberon looks pretty good. Let's click on that one and say we'll use Uberon, and actually, while we're in CEDAR, we can take a look at Uberon and see what Uberon says about tissues and make sure that this is actually the ontology we want to use. And if that's the case, then our middle panel is where we can actually start using that ontology to fill things out. We can have, for example, all of the fields of that ontology specified on the left. We can say, well, we want to fill in in this case, the tissue where the sample comes from. We can see blood, liver, bone marrow, breast, lymph node, lung, colon. These are all Uberon controlled terms that provide the basis for selection that will allow us to choose the tissue where our sample came from, and we can use an Uberon controlled term to make sure that we type in, for example, blood, and we don't misspell it, and we don't call it something stupid. What's also important is that we use the context of the information in the metadata window to be able to help the user do this much more quickly than would otherwise be the case. So for example, suppose we want to figure out what is the tissue, and that we say the tissue is brain, and this happens in Homo sapiens, and in this particular sample in a 74-year-old individual, then CEDAR, because it has seen this kind of pattern before, can suggest, well, look, for the disease involved, since we know the tissue is brain, maybe you want to think about a disease like Parkinson's disease or central nervous system lymphoma or autistic disorder or melanoma or whatever. And what you see is that all of these terms are coming from the disease ontology, and the number that you see in parentheses represents the frequency with which that particular disease has appeared in previous samples that have been entered through CEDAR that are associated with that particular organ. And so since we know we're dealing in this case with brain, we can see that 39% of our samples in this kind of situation have dealt with Parkinson's disease. Maybe that's a good place to start. We, of course, can choose any disease we want, but by taking advantage of the patterns in the metadata, we can more easily and more quickly enter metadata for very complex specifications. If we want to say, for example, that the tissue is lung, well, we get a whole set of different diseases in a different set of order. We can say that lung cancer occurs in 61% of previously entered samples. Chronic obstructive pulmonary disease occurs in 13%. And again, we can order the selections from the menu to take advantage of what we already know about previously entered metadata to ease the fashion with which new metadata get entered. And we believe that this approach will make it much more palatable to be able to specify complete standardized metadata. So again, our approach in CEDAR is to author the template, to fill in the template using whenever possible ontology terms, to save a copy of what we've just done in order to be able to reuse it for purposes down the road, and then to take our metadata and the corresponding data and put them into appropriate repositories. Now in CEDAR, what's really important to stress is that all of these windows that you've seen on the web are not canned. They're generated on the fly. And they're generated from all the semantic components, the template elements, the templates themselves, ontologies, value sets. All of these are managed as first-class entities in CEDAR. That means that the user interfaces and the drop-down menus are not hard-coded, but are generated on the fly based on previously made selections so that the program is able to provide as much information as possible up to date, and to do so taking advantage of the particular context in which those selections get made. I should note on the side that all of our software has defined APIs, so basically if you don't want all of CEDAR, if you don't want to have the web interface, you can still get CEDAR functionality over the web through our APIs. And as CEDAR generates all of its metadata in JSON LD if you care, so there's a standard way of talking about all these metadata, so it's not just some abstract arcane XML, but actually a standard that can be reused for a variety of purposes. Now, because all of the entities in CEDAR are first-class entities, because each of them is used to generate the user interface, if I wanted to, I could create a template related to earth science. I could talk about some experiments in earth science. I could have in Bioportal, if I chose to put it there, ontologies that talk about rocks. And although this happens to look a lot like the interface you've seen previously with CEDAR, here is the same CEDAR technology, in this case, acquiring metadata for an earth science experiment using an ontology of rocks, all of which can be generated in minutes, because all of the interface specifications that you see in CEDAR come directly from the semantic content that CEDAR operates on. We're testing this out with a variety of partners, and I should say that although we've been working on this for a couple of years, and actually it's been a fairly long haul, not only to develop the technology, but to ensure that this technology fits into the workflows of the people with whom we collaborate, we're in a state now where we're really starting to get some results. We're working very carefully with IMPORT, which is a repository of data related to immunologic disease. IMPORT basically stores all of the data that come out of experiments that are funded by the Division of Allergy, Infection and Transplantation of the U.S. National Institute of Allergy and Infectious Diseases. And we are now starting to use IMPORT in a regular way to be able to take CEDAR-authored metadata and store those directly in import with the associated data sets. Major collaboration going on with LINCS, which you heard about earlier today. LINCS is a large consortium of NIH-funded investigators, from all of whom are receiving support from the Common Fund, who are involved in studying cell signaling. We're now in a situation where LINCS investigators are able to do experiments, author metadata using CEDAR, and upload those metadata directly into the local repository that the LINCS folks are using in order to manage all of their data and perform their data stewardship. So the four-letter word that won't go away is fair. CEDAR has been designed to make metadata and their corresponding data more fair. And I think what makes it very exciting for us is that this kind of technology, we believe, is going to lead us to a situation where the metadata that we use to annotate data online will be more standardized, will be more complete, will basically help us in a variety of tasks that are important to us, including data discovery, data exploration, data integration, and secondary use of data sets. I think this will not only help us in being able to integrate data and put data to new purposes, but basically facilitate scientific communication in the broadest sense. And I think what's very exciting to us is to think about expanded metadata, not only as a means of making data sets better, but as a way of making the communication of science more informed and more appropriate, and ultimately provide a new mechanism whereby we'll be able to get more information about what is in our experiments in ways which will be useful not only to people, but also to computers. Let me stop there and let me thank you. And we have time for maybe one or two questions, if there are any. Yes?
So, a lot of work done showing that annotation is so much easier while doing the experiment. What sort of outreach so this is great for once you're submitting, that might be too late. What work are you doing to integrate upstream in the experimental process?
All right, so the question is, this is wonderful, but the timing may not be perfect. It may be either too early or too late, depending on where we are in the situation. I think one of the things that excites us a lot, not anything for which we're currently funded, would be to integrate CEDAR technology with electronic laboratory notebooks, so that at the time that protocols are first being identified, at the time the experiments are being done, much of the metadata that is already being written down in the ELN could be gathered and ultimately processed by CEDAR and submitted. The other thing, again, for which we're not funded, so it's not on our immediate task list, but something which would be easy to do, would be to look at the metadata in existing repositories that we know are supposed to be encoded using ontology terms, and use techniques such as the NCBO annotator service to be able to try to substitute text for ontology terms in a way to enhance metadata, at least with ways which may make the metadata then more searchable with tools such as BioCaddy. So we think there are opportunities both upstream and downstream to improve metadata, in addition to being able to make metadata better at the time that initial experiments get documented. Yes?
So Mark, I know we've talked about this quite a bit off the stage as well. One of the critical elements moving into precision medicine is the capability of getting all sorts of clinical data standardized. In this regard, if we use TopMat's data sets as an example, anyone who spent hours or days or weeks like our groups have done would see that each type of omics data sets in the TopMat has its own standards and units, and there must be like 10, 20, 30 different kind of things that's been all over there, all over the place. So what are your thoughts regarding begin a generalized omics metadata standard templates for clinical data sets?
That's a great question, Pepe, and there are a lot of dimensions there which I cannot obviously answer in the time we have. I think what is most critical, as you alluded, is that when looking at collections of metadata, what is very important is to be able to make sure that one does appropriate binning of data so that even in the absence of PHI, one does not have the ability to try to trace the source of data on the basis of extreme information which could be inferred from a very small sample size, which is probably one of the most important things to do. I mean, ultimately, each of us will be uniquely identified by our DNA. And so the problem then becomes, what information can we encrypt? Can we omit? Can we basically make available in some way which requires some secondary step to perhaps a trusted partner in order to reassemble? Those are really great questions to think about. Unfortunately, we have not actually considered those in the kinds of experimental work that we're dealing with, but obviously we need to deal with that as we move forward. I think we should stop there so we have time for our next speaker, who is